Good morning. It's really a pleasure to be in this fantastic place with this audience. And I was here last week for another meeting, and the inspiration to be on the podium was that now we need to take out our violin from our cases and start playing Mozart, because that's the place, that what inspired me. But today we're talking about something almost as exciting as that. And we're going to be talking about biomarkers. And please, my guests are three really illustrious scientists that come from academia, from industry. And I'm going to try to ask them what I don't know, because that is the pleasure of being a moderator. And, actually, the state of a moderator doesn't fit me, who knows me, so that I'm not very moderate. So I think that really gives me, some advantage in this thing. A small introduction. Biomarkers are really changing the way we think about medicine. But it's also a very dangerous, direction, because sometimes, these beautiful numbers that come out from our body give us the illusion that we can understand every physiological process in disease anticipation that we can get. And one story that I want to tell you is that, most of these analyses are done by these fantastic young people that are coding, use AI to make this, very, very complex algorithm and come up with solution and results that are incredible. And I was talking to one of them, a couple of weeks ago, and he was telling me that I found a combination of biomarker that will predict who will develop the disease of the liver. And I was joking, and I asked him, well, you're talking about the right liver or the left liver? And I thought it was a joke. But he looked at me very seriously. I don't know. I need to look into that. So I'll give you an idea of what is the dissociation sometimes between, working on number and, working on humans. But I'm sure that my, guests will be able to address some of these issues really, really well. So I'm going to ask them to introduce themselves. But before I introduce myself, my name is Luigi Ferrucci. I'm the scientific director of the National Institute of Aging. I've been working on aging my entire life. I am a physician, a gerontologist, and epidemiologist.
Good morning. I'm David Glass. I'm a vice president of research at Regeneron Pharmaceuticals. I started the skeletal muscle group there many years ago, and have more recently started a group on aging and age-related disorders. I also teach, I'm a lecturer at Harvard Med School. I teach a course on experimental design to try to improve reproducibility of published work.
Thank you. I'm honored to be here. My name is Peter Abadir. I'm a practicing geriatrician. I'm a clinician scientist that I work at the interface of aging biology and looking at physical and cognitive decline that happens with aging, specifically looking at frailty and early signs of Alzheimer's disease with some flavors of AI. I'm one of the PIs of the Johns Hopkins Artificial Intelligence and Technology Collaboratories of Aging Research, and I'm also PI of the Hopkins Older Americans Independence Center.
Good morning. My name is Vadim Gladyshev. I'm a professor of medicine at Mass General Brigham and Harvard Medical School, also associated with the Broad Institute. I'm interested in aging, so I have a lab that half experimental, half computational. We try to understand the nature of aging, how we can measure it, how can we interfere, and find any therapeutics for it.
Great. I'm going to ask the first question. And my first question is something that I'm thinking many times. What the hell is a biomarker? What is your definition of the biomarker? Because the definition have changed substantially over the last 10 years.
So I think it's an important question because there's been over the last 10 years, you might be aware, people have published on what they call aging clocks, which mean changes in either usually in your epigenome, meaning sites on your DNA, which they say correlate with age. And then over the years, the same people keep every year say, well, my old clock was bad, but my new clock is good. And they keep modifying these epigenomic clocks. And to me, what is a more relevant aging biomarker are things which change as a result of age, which, as you pointed out in your story, which are predictive of disease or a relevant phenotype, and which, if you perturbed that disease or phenotype, the biomarkers would change as well to show that you're doing something that's good. In other words, it has to not just correlate with aging, but correlate with perturbations of aging that are predictive, which is a much higher bar, I think, in terms of what a useful biomarker is.
I want to take that and push a little bit and talk about what the issue with a lot of our current model of building on specific biomarkers of one or the other, and using an example of some of the medications that are used in the market for treatment of Alzheimer's disease. And I'm not going to specifically mention the drugs, but some of them have on their drug insert a statement saying this drug is not intended to treat or prevent Alzheimer's disease. You basically start to see how, sometimes our gap is not really in the discovery, but more of the translational. How can we take those measures and connect them to meaningful outcomes to older adults? And I want to argue that, whether this is a blood and now with all the digital biomarkers, I think we need something that is more connected systemically. And I'm biased, but I would say something that, like the concept of frailty, is a systemic readout of biological age and can be a biomarker in itself because it's looking at the whole system biology and translating it into something that changes over time in patients. That's just a little bit of my thoughts on what constitutes, a meaningful biomarker.
Biomarker of aging is something as a parameter that measures biological age, in essence. Then the question is, what is biological age? Biological age is a state of the organism in the aging trajectory. And ultimately, in order to understand what is biomarker, we need to understand what is aging. And here is a problem, because we discuss here aging, but I don't know, but we might talk about different things. In fact, a couple of years ago, I chaired the Gordon Conference on aging, and I gave a questionnaire to people. These are all experts in the field, asking the most basic questions, like what is aging, what is biological age, is aging a disease, something like that. And there was no agreement on any questions, and over 100 people responded. There were 100 definitions of aging, and when I tried to classify it, so for some people, they equate aging with demographic aging, like, something ages if it's more likely to die tomorrow than today, mortality-based. For other people, aging is functional decline, something that becomes worse over time, functionally, like cognitively or any other function. For other people, it's just any period of the development or any age-related change. Other people think it may be damage accumulation. And so all these features, they kind of different agings. and so when we discuss, we need to define what exactly we are trying to measure here. Are we measuring like functional aging or like a physical aging, like that would apply to inanimate objects like cars and so on, or maybe biological aging that would involve accumulation of molecular damage? So, for example, if you ask me, then I would think that aging is something that starts very early, already during embryonic development, because organisms live, and the imperfect organisms, they accumulate damage, start accumulate. But during this time, function still improves during development. Later, in the adulthood, everything kind of goes down. Function is decreased, and damage still accumulates, and so on. And we can interfere at different processes and different time periods during this process, and also can interfere with different types of aging. And we need to define going back to the biomarker, we need to understand what are we going to measure. That's really the question. And I think it's a matter of discussion and debate because there's no consensus here.
I think this is a very good start. I think that what I notice is that all of you, when they think about biomarker, are thinking about biochemical, blood biomarkers. You know, I think that because most of the revolution has been there. So physicians have used blood biomarker forever. You know, when I graduated, and I don't want to tell you how long ago, because it was long ago, you know, we were using those 35 indicators, you know, do the start of every patient coming to the hospital, and we're still using those 35. What is the difference is that, you know, when you had to liver enzymes that were high, you knew what was happening. You know, there was, the liver was not working, and so there you had some indication of necrosis of the liver. Today, we have this complex biomarker. Most of the time, we aggregated 100 or even 1,000 of information from these multi-omics. And so, and we come up with, you know, indicators that are much less specificity that they had, those 35 biomarkers. How do we increase that specificity? How do we do better than that? Because that's really one of the key for clinical translation.
Well, I'll give you an example that's not a blood biomarker. So we have a paper in press where we looked at changes in gene expression in about 35 different tissues in rats and mice, and then correlate to humans. And just to give you an example of very useful biomarkers that taught us something we didn't know, we looked at so the Sprague-Dawley rats get sarcopenic. They lose muscle function the last third of their life. And you see these decreases mitochondrial function in the muscle, completely correlating with the decrease in strength and size of the muscle, which also, by the way, happens in humans. And so that's a biomarker of loss of function in the muscle, which is actionable. But then the shocking thing was we also looked at the spinal cord innervating the muscle, which has the motor neurons innervating the muscle, and the spinal cord was showing the same sort of changes before the muscle was. So this is a biomarker showing, and by the way, you get functionally denervated as you get older. The reason older people get like a functional tremor is you actually lose motor neurons. They retract from your fibers, and your resident remaining motor neurons send out innervation. That's why you start getting discoordination. And the motor neuron changes happen before the muscle changes. So these are biomarkers, but they're actionable because they teach us we have to not just worry about the muscle, we also have to worry about the innervation and the nerve. And like there's a chicken and egg problem of what comes first. So I think that gives you an example that as to how to be more specific, the more granular you get to the different processes and different systems, which like Vadim was saying, like aging is could be different in each organ, and you have to get to mechanism that shows you how you can be, understand, oh, this is a problem I didn't know about by following this, and now I have to think about how to drug that or how to perturb that.
So I think I work with, as you know, Luigi, I work with some of the digital biomarkers engineers, and it drives them crazy, the heterogeneity of aging. When you see one older adults, you have seen one older adults. One 70-year-old, you have seen only one 70-year-old. So that heterogeneity in the biomarkers add to that complexity that was just mentioned, that unfortunately we are not 99% clones of each other. We have exposomics. We have lived experiences that are different from one older person to the next. We have medications. And I want to just explain when I say digital biomarkers, and I'm sure the audience here is aware of many more than the example that I will use, but one of the techniques that we have been working on is using eye movements to identify older adults that have early subtle changes of cognitive decline. And the system basically asks you to read a sentence. And when you start reading, it looks at your eyes as you are looking at the words. You know how large language models is built on predicting the next words in the sentence and building that on that? Your brain works exactly the same way. You look at a sentence, you skip over words. You're skipping over the second and the third word, and this is a digital biomarker because older adults with Alzheimer's, early subtle changes of cognitive decline, their brain is already sensitive to that damage that they look at every single word. So when the computer captures that digital signal, then you start to identify as an early signs of cognitive decline. I think the issue with the complexity that now when you take all the multi-omics and give them all the digital signals that comes from the right liver and the left rival and right side of the heart and left eye and right eye and build them all, I think it becomes much more complex. But I think we really don't need more multimodal data or multi-omics, but need integration. All this has to be anchored back to what matters most to our older adults. And this is just what we talk about in geriatrics, and we always teach that the 4 M's of geriatrics, the mentation, mobility, medications, and what matters most. So we're not looking for more multimodal data, but more multimodal integration of this data with things that matter to our older adults, like gait, like speed, like sleep, like cognition. So this is how I see it. And I think AI is really, we are in a very good place to start that integration and not only look at the animal models, but take from the animal models and actually start to align what's similar and what's not similar in those biomarkers between humans of different races, ethnicities, and exposomics, and start to have more targetable areas. Thank you.
The question was how can we be more accurate? And it's a real challenge, actually, because the aging space is a very complex space, and any biomarker is going to be just one slice of it. It's not going to be very accurate. So even like composite biomarkers based on single omics initially, I mean, they have their limitation. And I would agree that we would need many slices, as many slices as possible, complex slices, and we could integrate omics-based or functional measures and digital and so on all together. The challenge is to kind of cover as much space as possible in the simplest way, and then, as Peter said, integrate this all into a model. But we have to understand that organisms they're kind of both have systems, autonomous and non-autonomous, kind of interaction of those, and they age, kind of they talk to each other, but also somewhat separate. So sometimes one particular system might become older than another. So like a kidney, for example, or heart or brain, and therefore we have to kind of measure that in a way with kind of multimodal way. So it's a challenge. So I think it's rapidly evolving now, this space, and it's very exciting to be.
Wonderful. I think that, so when the biomarker revolution started, people were thinking of biomarker in 2 ways. One is predictive. Biomarker predicting something that will happen later on. Very important, because I think that we can create AI can take care of all the people with disability, but if the accumulation of all the people with multimorbidity disability will continue rising at the same rate, there's no healthcare system that can deal with that. So slowing down this progression is important. The other one was the biomarker will give us some hint about the underlying mechanism that was you were talking about. But in the end, while the biomarker are now a nice method that the scientists use, and they publish beautiful paper, and we are all glorious about those papers published. The final goal is to put them in the clinic. because if we don't, it will be useless. So in your view, what is the thing that need to happen so that the blood biomarker will be included, will be used by physicians? How we can convince them that that's really a tool that can improve their capacity to care for their patients?
Well, as you said, the reason we've done certain tests for 50 years, like looking at liver changes, is we know what they mean. We know if liver enzymes are up, you have liver damage, and you have to treat the patient in a specific way. So the thing that we really need to do, and hopefully AI can help in this, is to correlate particular aging biomarkers to particular phenotypes, which are then actionable. Otherwise, it doesn't matter. Who cares about a particular perturbation if it's not actionable, if it doesn't tell you? So ApoE4 homozygotes, which the prior speakers were mentioning, which tells you you're going to get which is doesn't tell you, is highly predictive that you're going to get early Alzheimer's disease. If you're an ApoE4 homozygote, that's actionable in terms of at least the risk, but right now we don't have a treatment for Alzheimer's. But if we could either perturb the ApoE4 genotype or understand the mechanism there now so as to do something, that's like an obvious example of an aging biomarker that's predictive of a disease, which then you'd want to know, because then you could perturb the patient. I'm very interested in frailty and sarcopenia. So AI, we've just heard before, by monitoring you, if it starts telling you you're losing function, you're decreasing speed, and then people, we can give a medicine to increase muscle mass, and now I know we have to also worry about innervation. That's an actionable biomarker that can, from an AI point of view, say, oh, this person is at risk of sarcopenia, they're starting to slow down, or they're showing signs of muscle atrophy, we should treat them sooner rather than later. So I think the key thing in biomarkers is you shouldn't just measure things to measure them. It has to be related to a phenotype that either alerts us we need a medicine there, or we have one which now we can use.
So I agree 100%. I think those biomarkers, in order for physicians are really hard to work with, right? Because they will look for what is best for their patients and make their decision based on guidelines, based on their clinical practice, clinical experience, comfort with medications, etc. The risk-benefit is huge when you're dealing with patients. So I do think in order for us to give them a biomarker that they will believe in, that has to be anchored to something that they can do. I'll tell you that you can do a lot of genetic sequencing and come up with so many of those kits that are available in the market that will give you probability of developing something over this next 10-15 years. What happens to patients when they get something like that? You ruin their life, basically. You give them a sentence of death over their head. They keep thinking about that. They actually might develop the disease just because you give them that probability, not because they have a more likelihood of developing it. And insurance becomes even more complicated when you start dealing with that. And it's just a big thing to give biomarkers that are not, that you can't do anything about it. And I do think that if we start looking at aging biomarkers that are anchored to functions that patients can care about, that are and can be intervened on, it's going to make them adapted. I had a physician that was my mentor that invited me to his 50 school anniversary class of his MD school. And I talked a little bit about AI and the ability of AI to predict subtle early changes. And he used the example of himself, saying that last month I fell and I was not told that I was getting weaker. I went to my physician, he put me on physical therapy, and I have not fallen since then. So there's something to do there. But why couldn't my physician tell me that I was getting slower or weaker or define something that I could do early on. The problem with the clinical visits also is that it's hard because it's limited time and you do course measurements. So I do think when we think about biomarkers that will be connected to something you do, and we have tools, we have physical therapy, we have nutrition, and I know that there are a lot of frowning on protein intake and all of that, but the problem with what we do now is that we wait until there's a full-blown disease and then start thinking about nutrition and exercise and all of that. So maybe AI and integration with those multi-omics will allow us to shift the biomarkers to much earlier sensitive point where physicians will adapt it, because now you can start having some very specific interventions. Long way to say that you have to give them tools to, if you get this, you have to do this kind of thing.
I also agree. I think maybe just to add that such a biomarker would need to be validated in clinical trials and hopefully get FDA approval. Just one example, I'm just excited about this program from RPH called PROSPER, and the idea is to develop new type of biomarkers for intrinsic capacity that would be used across the lifespan and would be very inexpensive and easy to use, like could be applicable at home testing for about $100. So that's kind of the maybe immediate goal, but with the understanding that in the future it's going to be rapidly replaced with some kind of large health models, presumably. Yeah.
So I think that the three of you, in one way or in another, work on biomarker. They do research, they try to understand more. So if you had kind of a dream, what would be, the technical revolution that will be required to really improve the utility of biomarker, in care and also in research, but especially translational in care? Is this a technical improvement, or is what we already have good enough and we need to handle that better?
Well, my job is to try to develop new medicines, not just biomarkers. So for me, a biomarker is most useful if it teaches us like what molecular process is being perturbed in a disease, such that we can develop a medicine to treat that disease. So it's a more, I think people sometimes get bored when they hear biomarker because they just think it's a correlative thing and doesn't matter. But if you use it correctly, and this is where, by the way, AI, which is the topic of this conference, can really help, is like if you can integrate, for example, at our company, we've sequenced over 3 million exomes. So we have huge amounts of human genetic data, which is itself, which gives you markers of genetic perturbations that make it more or less likely to get different diseases, and then we can integrate that with healthcare records, and then integrate that with omics and all this. So when there's this huge amount of data, that's when AI can be extremely helpful to, if you ask the right questions, to get, to understand correlative evidence that then allows you to see if perturbations can help. So for me, it's not just biomarkers to go into the clinic to sort of correlate with something, but it's to also teach us how to make, what's the molecular changes that we should drug so that we can cure or prevent or at least treat disease. And then, of course, once you can do that, then you don't have to argue with people to use those biomarkers, because then those are the molecular criteria to use a particular medicine versus not. Like, very simple example, high cholesterol. So which is a marker for getting heart disease and atherosclerosis. So we use, for 50 years, whatever, use statins, right? Or 30 years. And then, and now we can use PCSK9 antibodies that dramatically decreases cholesterol. And the more markers you have for failure, the more criteria you have to use one medicine or another. And that's a very powerful example that you don't have to teach people to use that biomarker because it's clear, to your point, what do they do. So they have an X, they measure X to do Y. So I think if it's a point of like trying to convince people, no one's going to do it because doctors don't do what you ask them to do. I'm an MD, I can say that, But if they understand they can prescribe a particular medicine, or from our point of view, research a certain area based on a biomarker, then you don't have to convince them.
So if you allow me, and sorry, I think that you touch on one aspect of biomarker, the biomarker that is specific of a molecular change that can be identified. However, there is another face of the coin, which is the biomarker that we don't completely understand, but we know that are predictive of something from large epidemiological studies. So I would like the other two, speakers to touch on this second way. What is the dualism there? Do you think that what we don't know now, we will end up knowing, or the biomarker will be useful regardless of whether they tell us something mechanistic or not?
I'll borrow from Deepak, this language, Deepak from UMass AITC, that bad data is better than no data. So I do think that what we don't know is still good to have some partial knowledge of that biomarker and work in the future. But I want to just build on that also and say that I think the problem with biomarkers is a little bit more complicated than just what biomarkers we have, because I'll use an example from a paper that we all have been working on and became an area of argument. Taurine has came as a biomarker of aging, a science paper, very famous science paper. Another very famous also science paper came and said taurine is not a biomarker of aging, and it was a heated debate whether taurine is really something or not or nothing, and we were all discussing and debating whether the difference is because it's measured on different machines or different systems. So I do think the biomarker problem here is a lot of it still is in the systems. So the shared protocols, the measurements standardized. When you tell me that a CBC, a hemoglobin of 5 in an older individual, that is a 5. And if we do it at Hopkins, it's a 5. If we do it in Texas, it's going to be 5. The problem is that we're measuring biomarkers once, reporting it cross-sectionally, and continuing to looking at that. So I think that if I have a dream, is that the biomarkers become more standardized in terms of how you measure them. So shared protocols, open database, longitudinal measurements of those biomarkers, so that eventually we develop the model that can tell us what can we do differently for our patients, including the biomarkers that you mentioned, Luigi, that currently are stable. We don't know why they are stable, and we think as long as they don't go up or down, that is not connected with function. So I think longitudinal and then standardized is what we need for biomarkers.
I think roughly everything changes with age. So it can be measured in so many different ways. Just from my experience, we started with epigenetic clocks and transcriptomic clocks, proteomic clocks. Now we can measure aging from like MRI imaging, retinal scans, from voice, from videos, locomotor activities, and kind of integration of all this. Ultimately, it's all needed because they much better represent that complex biological system that humans represent. And any slice of it, like any single biomarker, would never be accurate, whether the taurine or whatever, muscle function, none of that. It needs to be kind of systems kind of view on the organism. And ultimately, this is needed to develop therapeutics, which we don't have against aging. And to me, there could be 3 types here. One type is where we could kind of slightly remodel the organism. This is kind of, Ozempic, metformin, rapamycin type. It would be very little benefit. So maybe, 1, 2, 3 years maximum. benefit in humans. But this kind of multimodal view on the human organism also would tell how a particular human ages. So somebody is more older, for example, in the brain. So in this case, we would need to have personalized intervention early on, before symptoms appear, before functional decline, for that brain, specifically for brain system in that organism. Somebody is older, in the liver, so there's going to be a different interventions. Ultimately, the majority of interventions would need to be personalized. That would be the most impact in terms of health span for humans. And that requires kind of multimodal view on that particular organism. So it's very challenging, very, very difficult. We are not there yet, but I think ultimately we'll come there. And I would mention the third type, which is like we are not even touching yet, actually slowing down the aging process. It's like, for example, when we compare across species, like compare mouse to humans, there's a 30-fold difference in lifespan. So nature can easily change lifespan across species. We are unable to do it for now. But if we learn how nature does it, maybe we could also interfere with the aging process itself, kind of slowing down, maybe achieving radical lifespan extension. We don't know how yet, but that's the goal.
That's absolutely great. I completely agree with you. I think that there's evidence in evolution that age is malleable, can be modified. I think that it's probably very complex and tricky, but hopefully we'll get there. But I'm going to ask you a practical question that is related to that. Maybe you can start, we can go in the inverse direction. So, we have several biomarkers of biological aging that we're using. And there are two tricks with those, I think. One is you don't know whether changing those biomarkers really change biological aging. I think that they track biological aging, but we know any evidence that the contrary is true. And the second thing that you actually taught me about is that some of those biomarkers are, indicator of damage, something that causing the disease and the outcome. Some others are certainly attempt of the organism to react to pathology, to defend itself, resilience, indicators. How do we work this complexity? What do you think about can be done to address that?
Yeah, it's a very important point. So I think what you refer to is causality, like a causal biomarkers, ultimately, yeah? We don't have causal biomarkers yet. So a couple of years ago, we published this paper we call the causality-enriched biomarkers, where we identified CPG sites which are more causal. Kind of partially addresses, but it's still only one layer, only epigenetic layer. So I guess there is work ongoing now. It's not yet published on kind of proteomic causal biomarkers and maybe in the future. Because yes, we need to quantify damage, which is very difficult because it's much of it's stochastic and there are no good methods to measure it. And there is also neutral changes, but also kind of adaptive changes, but because organism responds to it. And so if we interfere, if we have a biomarker, like not causal, and we interfere in therapeutics, because maybe we can interfere with this adaptive response. We kind of destroy the protection. And so based on the biomarker, it would look like it's beneficial, but actually it's not. So this is a real problem with biomarkers.
I still have a mentor that used to hate two words, causality. So he said, never write it in any paper and give any hint that you are going for causality. And then the other one that we proved. You never prove anything in any paper. So don't use the word proof or causality. But I do think that the word causality is a big word that, yes, up to a certain stage, we never had the ability to get that inference of causality. I think AI can help us. But I do want to build a dream here that I do think many of you have kids, many of you have gone to their kids' appointments and seen growth charts. Pediatricians have pediatric growth charts. They put your child on a weight and head circumference, and they keep telling me your child is in a 5th percentile of weight. And I keep feeding him butter and food and all of that, and still the 5th percentile. And they were happy as long as you stay within that trajectory. I think in aging, we need something like that for those biomarkers, that you are building them longitudinally. And as long as you are in the 5th or 95th percentile, it doesn't matter, because people are different and we will have different levels of those biomarkers. But a change is really what's going to make a difference. And finding out when that change happened and start putting some thoughts on what does that mean and putting interventions, I think is one way to act on. So I do think this is where using the multimodal longitudinal integration and looking at AI to use that to build the model for us. And I want to say that if the AI is going to be only telling us that they are aging, that it's not complete. Any model that cannot tell us what to do differently for that person is an incomplete model. So we need those models to be able to pick in the biomarkers, but to tell us what to do differently based on those integrations. And I forgot to add it, the biomarkers, wearables. Wearables are extremely rich set of data at home that actually is more the truth of what happens at home. So this is a set of omics that we don't think of a lot, but that integration is going to be important also as we build the multi-levels omics that only AI can put together for us.
I'll give you an example of a protein that goes up in age pretty reliably. Obviously, it's a probability thing, but when you look at thousands of humans, this thing goes up. It's like GDF-15. GDF-15 turns out to be pro-anorectic. So when you have cancer also, and you have cachexia, this thing goes up. Pfizer did a clinical study with GDF-15 settings of cancer cachexia and showed that you had significant efficacy in terms of maintaining muscle mass, which is important because then you can continue to take your anti-cancer medications. So here's an example of an aging biomarker where we understand what it does, which they created an actionable clinical trial which had actually a positive benefit on patients. Also, it turns out for frailty, if you have high GDF-15 and people simply aren't eating as a result because they become anorectic, that's another reason you lose muscle mass. From a trivial reason, you're just simply not eating protein. So you're sensitized for that. So that's just a biomarker story. But then where does AI come in? So why does GDF-15 go up? What's the molecular mechanism? Can we understand genetics of people and then correlate different genetic backgrounds to people who have more or less of this one biomarker? Now multiply that by 5,000 biomarkers. And now you see where you would really need AI to integrate all of this data to then see what really correlates with. Do you see what I mean? I mean, so that's an actionable aging biomarker where we have an actionable mechanism where you can imagine complexifying it even further to all of our data, and then you really see where AI starts to come in to be quite helpful.
So I have another question that really is kind of a question mark in my mind. I think that if you think of 10 years ago, the biomarker that everybody was looking at were the inflammatory marker, the cytokines. It was the era of the cytokine, the IL-6, the IL-1, the TNF-alpha. Everybody was measuring them with the idea that if you block inflammation, you can promote resistance and, prevent chronic disease. Now, this exploded at the time of the CANTOS trial, where, the inhibition of I1 beta, was followed by a reduction in cardiovascular event. But enthusiasm was diluted later on because there was a singularity. The trial blocking inflammation in chronic disease worked there, but didn't work in many other instances. So there was a number of failures, and the interest on modulating inflammation is kind of fading down. Why do you think that happened? Why do we see that inflammation is such an incredible biomarker of aging predictive of many diseases, but blocking inflammation does not work.
Well, I wouldn't go that far. I think that we want these binary solutions. And this is also from, people wanting to publish in Nature, and then it's like, these editors say, oh, you want these magic things that do everything. So it's very clear settings where inflammation is harmful. I mean, especially even in Alzheimer's, microglia become much more involved. For muscle atrophy, cytokines dramatically increased muscle atrophy through NF-kappa B activation is very clear. One of the problems is again, it's not binary, because for regeneration, you need like a burst of inflammation for the muscle. Like if you work out, you get this burst of inflammation, and then you have to have it go down, and then stem cells can then differentiate. But if you have chronic inflammation, it gets blocked. So it's more, biology doesn't tend to be binary in black and white. So you had the real stories are always more nuanced, which nature and science doesn't want to hear from. That's why you end up with the real papers are in JBC. So I wouldn't say that it's incorrect. Aging, I think, is a disease of inflammation. I mean, in many instances, you just have to understand where you can intervene and to what degree. Also, from cytokines, you end up playing whack-a-mole, because if you hit one cytokine, there are 15 others. So you need to go downstream to like JAK-STAT signaling and all of this. So I wouldn't say we're done with inflammation. I just think we have to be more nuanced and more focal in the particular settings where you want to perturb it.
So I 100% agree, and I do think that the story of inflammation is not yet completely explained, and I'll use a couple of things. I have been working with all those common targets, IL-6, TNF-alpha, TNF-alpha receptor 1, and we get excited about one cytokine or the other. But I'll use an animal study that we published to just explain the complex nature of some of those cytokines. So, Luigi, you're aware of our IL-10 knockout mouse model. This is a mouse model that we created to simulate frailty. IL-10 is the brake on inflammation. So when you take it out of the animal, they develop this low-grade chronic inflammation, which includes IL-6, TNF-alpha, etc. And they develop like frailty-like phenotype. They become kyphotic, they walk slower, they die sooner, and all of that. So we thought that, well, why don't we play with genetics here? Why don't we knock IL-6 on top of knocking IL-10 in this animal model? So we created the double knockout. With the goal, the dream, is that now you can reverse frailty and you can make the animals live longer and fix the issue. I mean, do you know that Max Planck said that the greatest tragedy of science when, data does not support a beautiful hypothesis? I think this is one of my moments of, this is such a beautiful hypothesis. The animal showed some improvement in the physical and motor functions, but guess what? Had 4 times mortality. And the editor of the journal loved that it was a supplemental figure showing mortality increased in this animal model. And we published it under the title, The Two-Phase Nature of IL-6. So that's the problem with some of those biomarkers. It is, they are part of the human biology. Getting rid of them or knocking them down continuously, I do think that this will not work. The questions for those biomarkers that need to be answered before they become actionable is that when, how high, and how much to treat and to bring them down. Because there is evidence that there is pulsatile treatments, there is longitudinal treatments, and there is, when do you start that? Are you talking about middle age? Are you talking about younger than that? Or are we also talking about only when you hit 65, this is when the time to start looking for biomarkers? And so there are a lot of questions, but I think it would be for us not the right thing to say that, this has been tried before. I think it was tried, but I don't think it was completely explored in the sense of the human biology and when and how much to give. Sorry.
Yeah, I will brief because I agree, and inflammation consistently come up in the data, but of course not always, because there are other systems that age. I would mention a kind of related issue, and actually the other day I was reading your paper on this infections, number of infections correlating with shortening lifespan. Yeah, that was very nice, because historically, we decreased the number of infections over the last 100 years because of antibiotics, better sanitation, and so on. And this kind of dramatically changed the population. So we kind of removed the factors that shorten lifespan. And now, on average, people look younger as well. So this is kind of related.
So I will have thousands of questions for you, and it's so entertaining to listen to your answer. But I want to give the chance to somebody to ask a question from the audience.
I'm a geriatrician and a neuroskeptic. I think the issue of longitudinal data is really important. When I was at Hopkins, I worked with the NIA, multiple longitudinal studies, so I think that's really important to look at these markers over time. But we've emphasized the heterogeneity of Alzheimer's. I think Vadim said, you've seen one older person, you've seen one older person. In the Alzheimer's field, we say the same thing. So Dr. Karlowicz, for example, suggested that we're going to have a panel of biomarkers, because one isn't going to do it. And those will be matched up to a cocktail of drugs. But we have not yet separated out what the heterogeneity of aging is in the brain and the heterogeneity of what we call Alzheimer's. And as you said, Peter, sometimes if you say to somebody you've got a marker for something, people will say, oh, I'm going to get it. And you really do more harm. So I guess my question to you is the biomarker business, and by the way, I'd emphasize the business side of this, we haven't done that, going to help us sort out this issue of what is aging and what we call a disease? Or is this more a matter of sociology than biology?
I think we have to do it, because to your point, if there's multiple mechanisms that can get you to Alzheimer's, by the way, even people with ApoE4, there's a couple of centenarians with ApoE4. So homozygous, so it just shows there might be a secondary mutation for those rare cases.
Or that they were exposed to an environmental toxin. We forget the exosome often.
No, exactly. So to your point, you have to get the data to then be able to answer your question, because that's the way to know, do we give one medicine or another? I mean, the heterogeneity demands much more granular sets of biomarkers to be able to sort through that.
I can quickly say that, like Bright syndrome, started, everybody with kidney failure was called Bright syndrome, ended up figuring out that there are tons of different kidney diseases that can be that are treated very differently from one another. Frailty is not immune to that. I think in 15 years, we'll discover that frailty was actually 15 different kinds, and that Alzheimer's disease is 20 different types that we are now trying to treat with one thing. And I think the future is for that personalized precision. I think it is for us. It's on us to do that, because the last thing I want for our patients, and I'm a skeptic like you, is to end up with another anti-amyloid antibody treatment that is targeting a brain biomarker that God knows exactly what are the side effects of those drugs, going to 20 or 40% of microhemorrhages in the brain. So,
just very briefly, I'm just very excited about biomarkers of aging, because I think it's one of the most important developments in the field. If we want to interfere with the aging process, we need to measure it, right? And now we're rapidly increasing our capacity to measure. That's the beauty of it.
And of course, some people think we can cure aging.
I mean, one small comment. I think that, I completely agree with you. I think that oncology and rheumatology have done incredible progress by, doing molecular characterization of profile of individual to sort out the heterogeneity of the different syndrome. And I think that needs to happen. We have another question. That will be the last question.
Thank you. So when you talk with an older person, they often say that what they want is health span, not lifespan. So what you're talking about with treatments is really increasing the health span of a person. So I'd be interested in your comments about what the difference is between increasing health span and increasing lifespan, which can be a very different thing, or it could be the same thing.
Well, for me, if you really understood life's aging and you fundamentally perturbed aging mechanisms writ large, you would do both. You would increase lifespan as a result and health span. I mean, Vadim studies evolution, and you have these amazing examples of like rodents where you have like mice and rats that live 2 years and squirrels that live 16 years. So and of course squirrels have 8-fold more health span, not just lifespan, because just evolutionarily they change. So, I mean, for me as someone who is interested in aging, I don't think those two things are as distinct as, to me, I think the health span point came up when someone had a hypothesis that was falsified about increasing the lifespan of an animal, and they said, oh, but the health span is better. So once you talk about health span, it could be anything. Do you know what I mean? If we're actually interested in aging, then I think I wouldn't dissociate those 2 points, because I think if you really can improve someone's lifespan, by definition, you should, in a way that changes aging biomarkers as opposed to just keeping someone on a respirator, you would improve their health span. Or at least that should be the criterion of a real lifespan perturbation, is that you are increasing their lifespan by delaying the things that happen when you're older.
I want to say, so I am deputy editor of the Journal of American Geriatric Society, and we have just recently announced JAGS-AI, so a new AI section built on technologies. And we're looking for, submissions and articles. But we opened with an editorial that I co-authored with our Dean of School of Engineering. And we quoted him saying that our mission is to put nursing homes out of business. The chief editor pushed back, saying this is offensive, you should not have this language, and all of that. And we pushed back and said, well, who aspires to be in his last years in a nursing home? I think quality life, free from disease at the end of life, is an important outcome. And I do want just to say that if in 15 years we're still sitting in another conference here and talking about improvement in survivorship with any of the interventions we do, I think we have failed the whole community. I think we should be looking at health span as outcomes and building in some of those functional measures in our clinical trials, looking at aging.
To me, there are 3 related terms: lifespan, health span, and longevity. Lifespan is how long we live. Longevity, whether we live long compared to the population, and health span typically viewed as like disease-free living. And so ultimately, we want to interfere with all of them, but sometimes interfering with health span from the disease perspective makes organisms still frail. They live long, but they're very frail. So I think I would agree with David here as also it really depends on how we define this kind of health span term.
Thank you very much. Give an applause to our speaker.